sizes of some of your plant parasitic nematodes. This diagram shows a really good spread of the different nematodes that we have. Up here at the top we have your biggest ones, like the needle and the sting. And the needle, you notice, is this big one that's folded over. It's actually that much bigger than all the rest of them. Down here you're going to see the lesion, which is a lot smaller compared to the other ones. So noticing the different sizes, these bigger ones obviously need a bigger pore space to move through. Those are going to be the ones that you find mostly in your coarse textured soils, the sandy soils. You hardly ever see these in a finer textured soil because they can't move through those smaller pore spaces. So these lesion nematodes, or some of these other smaller ones, you can find in any soil type. They fit a lot better in some of those smaller pore spaces, but you can find them also in the coarse textured sandy stuff. Looking over here, you'll see the SCN, the adult female there. You can see the relative size compared to some of these other ones. If you've ever seen an actual cyst on the end of a soybean root, you can see those with your eyes. But compared to these other ones, you would think you'd be able to see those too on the end of a root. The difference is that your SCN nematode, that hard casing it has, not only is it a thicker cuticle, it's got color to it, it's white or a yellow color, whereas most of your nematodes are going to have a clear cuticle, so you can't see them at all. The only time you can see them is under a microscope with some light shining up underneath them. So that's why even though these are such bigger nematodes, you won't ever really be able to see them with the naked eye. So we'll go over some of their feeding habits. These nematodes these are all common to the Midwest, and these top ones here are all ectoparasites, or they feed on the outside of the root, as you can see in the picture here. They'll just kind of move along the root and stick their stylet in and out, feeding on the outside. Some of these down here, the lance and the lesion, which is that smaller one I showed you earlier, they get inside the root and they move around, which causes a lot of damage when it's burrowing in between those root cells. So the important thing to take note of is when you're looking at nematode samples, you want to make sure that you're getting both the soil analysis and a root analysis. So we'll go over some of the relative damage risks you can see associated with these nematodes. Your higher risk nematodes are going to be those really big ones we talked about earlier, the ones that are in the sandy, bulky soil, the sting and the needle. They both have a really big stylet and a really big nematode. So the big stylet can cause a lot of damage to your roots, so the damage threshold for these nematodes is relatively small. 1 to 5 in 100 cc's of soil is a pretty big deal. When you get down to these moderate risk, the stubby root, the lance, the lesion, those are your two endoparasites here, they can have significant yield loss depending on what your population density is. So it kind of depends on what your population is for these, but they can be a pretty high risk too depending on the amount that you have. Down here we'll have something like the dagger, the stunt, the spiral. Those are a low or undetermined risk. Sometimes you can have lots of these in your field and not have any damage whatsoever. There's not as much data on these nematodes and how they affect different host crops and their damage that they cause. So that's why we put it in this category of like a lower or undetermined risk, is we're not really sure if they're causing any problems at all or very minor at the most. So if you're looking at your field and wondering if you have nematode problems, unlike some of the other pathogens, there's not a lot of things you can point out and directly say, oh, that's a nematode problem right there. You'll see chlorosis, similar to this picture, stunting. You can see these little tiny plants compared to the regular sized ones. Stubby roots or heavily branched rooting, or even root death. And as agronomists, you all know there's a lot of things that can cause any of these. Water, too much water, not enough water, pH imbalance, nutrient deficiencies, any of those things can cause some of these pictures. So you can see all the dead corn in the middle. So if you were going to go sample this, you don't want to go right in the middle where all those dead plants are. Most likely, you're not going to find anything in there. You're going to want to look on the outside edges. You want to look where the nematodes are actively feeding. If you go in here, either the nematodes have died from lack of host, they have nothing to feed on, so they've died, or they've tried to move around a little bit, move down. Some of these nematodes will move deeper in the soil with the roots, so they move down looking for stuff. So if you go in and probe with an 8-inch probe, you might miss a lot of them. So you want to go wherever those nematodes are actively feeding on the outside ring. That's where you're going to find those. This was a study done by Ann McGoodwin at the University of Wisconsin, just to show you sample timing, what might be recommended. We looked at 2 different nematodes there, the needle, which is one of those big high-risk ones, and then that root lesion, the little one that can go inside the roots. And they looked at 2 different times, right after planting and later on in the season. And they also looked at different depths within the soil, up to 15.5 inches. What you'll notice early in the season, you'll find these big needle nematodes in the top 6 inches, maybe 9 inches of the soil. But later on, most of them have moved down. They're moving down with the roots. So if you only probe down to 8 inches, you might have missed them completely and won't know that you had a problem. If you're looking at the root lesions, you'll notice most of those are in the top 9 inches all throughout the season. So you can see those anytime. But if you're still sampling later on trying to find a problem, like a lot of people will sample after harvest, they've noticed they had a yield problem, so they'll sample then. We might have missed all of these big ones that could be causing a problem. You might not notice that you had these in there. We recommend sampling real close to that plant and go at a 45-degree angle through that root system. You're trying to collect soil and roots so we can look at both of the different feeding types on those nematodes. If you're collecting out here in between the rows, we might be missing the ones that are actively feeding on the roots right there. Probe through that root mass, collect some of those root fragments. That's how we process those. And keep your samples cool. Don't ever freeze them. Don't ever stick them on the dash of your truck. A lot of these nematodes, for the root analysis, they have to be alive to be extracted. So if you freeze them, they die. If you bake them on your dash of your truck, they die, and we can't pull anything out, so we won't have anything to tell you. You want to collect over 2 cups of soil. Most of these extraction processes take at least a cup or a little bit more to make sure that we have a good representation. You want to take it randomly kind of around your area. Don't just take it right here. You don't want 2 cups of soil right here. You want a nice representation of what's around that area that you're seeing problems in.
Poncho votivo. Votivo, of course, being the nematicide. And this one is altogether very different from Evicta. This one is a biological compound. It's Bacillus firmus. That's a bacterium. This bacteria was isolated from somewhere in Europe, and this specific isolate, I-1582, is specifically what they're using. This is from Behn Crop Science, and me and several other nematologists have spent a significant amount of time with Behn at their headquarters looking at some of their data and their results, and none of us have a really strong feeling. We don't know how it's working. We just know it seems to be working, okay? We don't know the actual mode of action at this point, but it does seem to be working. That is a plant growth promoting not a rhizobacterium, but PGPR, plant growth promoting rhizobacteria, I guess. So this is one that would naturally occur in the soil anyway. So, okay, next one, Acceleron HX209. Undoubtedly, many of you will recognize the word Acceleron. Acceleron is Monsanto's combination of seed treatments that are available. Now, this particular one, the HX209, that's the one that's specifically a nematicide. This one in particular is a harpin alpha beta protein. And this is completely different from the other two. This protein is a naturally occurring protein that we know in other higher value crops. Although they have inconsistent results in some of those other crops, they have shown that by applying the alpha-beta protein, the harpin protein, that it can induce a systemic resistance in the plant to multiple pathogens and multiple pathogen groups. And so that's what's behind this reasoning. So by applying this, we're hoping to get some kind of systemic resistance. The advantage, if that's going to work, is that that protein does not have a direct interaction with any nematodes, and so there's no pathogen and pesticide interaction, so the chance of developing resistance is 0 to none. So in the first location, the upper left-hand corner, this is in 2006 in North, and at this particular location, we have tremendous nematode pressure. A lot of lesion nematodes, a lot of lance, stubby root, a zoo basically out there in the soil. That particular farm is irrigated and averages about 180 bushels per acre. Mixed soil types on one part, we've got a heavy loam. On the other part, where the nematode pressure really is the worst, where we are, is a sandier soil. And so that's where this trial was conducted this year. Now, looking at the treatments, I think you guys will be able to see that. Back row. You guys see this? This one's fungicides. That's your seed treatment fungicides. This is your seed treatment fungicides plus Cruiser. And then all the other treatments to the right are the seed treatment fungicides plus Cruiser plus one of 4 different rates of Evicta or Counter. Okay? And at that time, I think we were still using Counter 15. Okay? Now we're up to 20G. Okay, same active ingredient, turbifos. Now, in This particular year, you'll notice that we had a very strong dosage effect. With the addition of Cruiser to the seed treatment fungicides, we had an increase of just over 10 bushels. And then we increased it about almost another 10 with the addition of the low rate of Evicta. And you'll notice the highest yielding ones are all the ones that had the nematicides, whether it was any rate of Evicta or counter. Very strong dosage effect. You'll notice though, I don't have any letters above any of those numbers indicating there was no statistical difference that year. Okay, so that basically means there was a lack of consistency between the rep data. And everything we do is replicated 6 times in the field. Okay, and these are small plot trials. So this looks really good, at least that year, this environment under the heavy nematode pressure. Okay, at that same location, let's go to 2010. All these others are 2010 data, so I can try to show you the most recent data that we've got. In the Votivo trial, this is a Bayer-sponsored trial, on the left-hand side it says check, but that's a combination of all 3 seed treatment fungicides, plus one of these combinations, either Poncho 250, low rate of the insecticide, or Poncho 250 plus Votivo, or so on. We've got counter here, and then we've got Evicta Cruiser, Poncho 1250, your higher rate, or Poncho 1250 plus Votivo. To boil it all down, I'll remind you here, our 3 highest yielding treatments in that location that year were the ones that did have nematicides with them, whether it was the Votivo or the Evicta. So they all performed well in that case. Keeping in mind though where you're at. You're back in that same location in North Bend with high nematode pressure. Okay, so that makes good sense and gives us even more confidence about the products and how they're working. In another part of the field or in another location with lower nematode pressure, you don't necessarily see the same consistency or the same trends. And so that's why we're looking at multiple years. And so we're feeling more and more confident about the products in high nematode pressure areas. Now, we've been making a concerted effort to put the products in areas where we don't have significant nematode pressure. One of those locations is in that upper right-hand corner at Clay Center. And although we do have significant lesion nematodes in some areas, not necessarily in this location. I want to show you this. Also yield data, our check right here, that's with without any inoculum. Now you'll notice this says inoculated check. And so by adding fusarium, that's the seed treatment fungus fusarium, we knock the yield down over 20 bushels. You'll notice here, these are statistically significant. That was consistent across those reps because it has different letters above it. Now, adding fusarium obviously did a great job of causing disease. Well, we added fusarium also to these other treatments over here. But when we did that in the presence of the standard seed treatments, the insecticide, Cruiser, and the old fungicides, Maxim, Apron, and Dynasty, we increased the yields over 30 bushels. And that was statistically significant over there. Now, the new products that we were evaluating in this trial, Maxim Quattro, these are the same seed treatment fungicides plus an additional one, thiabendazole. Thiabendazole has activity against fusarium, which is why we were testing it in this environment. By adding that one, we had another 10 bushel jump, although that was not statistically different here. Coming over here with the addition of Evicta, another 5 bushel jump from the one before, again, not statistically different.